and welcome. Now easy. It's the Blue Stones. That's not for till another day. So, welcome. It is, it's functionally a Friday, right? It's Wednesday. We're not going to be here Friday. Today, Smith has compressed 3 lectures of material, extracted, excluded. It's been a really kind of difficult day on Tuesday of what not to talk about, because today we're talking about hexapods, which includes insects, and those are dear to my heart, and so figuring out what not to talk to you about has been a challenge. And first of all, there was a request on Monday to adjust some of the coloring and the word matches into this. So now, let's say we're here. We're all here. I know we're not all here. Some of you are still arriving. Hello. Those of you who have been here, you've had a chance to search out the Queen. Annotate now. Where do you think she is, if you found her? One hypothesis, 2 hypothesis, 3 hypothesis, 4 hypothesis. I am of a 1970s generation that can't do that without laughing like the Count afterwards. Mwah, Thanks, Count. So, everybody, you all got in the right neighborhood. Some of you did, in fact, find her, because that is her right there. So, well done you. That's harder than Waldo. That's the Queen Bee. And not, I suppose we should have played Beyonce for this, but the theme for the starting music, if you haven't noticed it, is Canadian artists and trying to be representative across Canadian artists. In this case, the representation was in age. This was the oldest Canadian singer that we heard. That was William Shatner, Captain Kirk, singing Pulp from his album Has Been. All right, so let's take this off. Thank you for your drawings, and I'll show you. There. She is there. If you like this kind of thing, there's actually an entomologist, a honeybeeologist, apidologist, who posts these kinds of things on her Instagram account. Queen spotting, she calls it. So you can search it by hashtag or by user. But there's another one right there. And probably now you're like so zoned into this kind of an occupation that you're like, oh yeah, I see it. There she is, right there. So, honeybeeologists, apidologists, here's where you go for your Finding Waldo. Now, before we get going, happy birthday, Sherry. And to the rest of you, we want to say that we know there's some questions about the exam. We're using the time that is in, or the date that is in WebAdvisor. That's Monday, April 26th. And it's not going to be starting at 7 p.m. because that's kind of ridiculous. And because it's on CourseLink, we're going to be making it as accessible as possible. So it's going to be open for 12 hours, 8 a.m. to 8 p.m. I'll even change that to 10 p.m. It's going to be open for more than 12 hours, 14 hours, during which time you will have double the amount of time to do it. It's usually a 2-hour exam, so we're going to give you 4 hours to do it in. And those are the details about the exam. It's going to be slightly different in that there are going to be a couple of true-false questions, but there's going to be more than that. There's going to be multiple choice questions and some short answer questions. So that's what to say about the exam. It's still like an impossibly long way away into the future. But today, let's get underway, and we're going to talk about hexapods. So, of course, we are within the arthropods. We've been within the arthropods all week, even into last week, and we're going to stay there at least until Monday. Monday, when we're going to talk about some Demodex and talk about that last assignment about your facial mites, because we're going to share some data and ask you some questions and kind of explore them together, and then send you off to answer some questions that I will write over the weekend. So, when we're talking about the hexapod, we're talking about insects and their close relatives. So they have uniramous legs that contrast that with the crustaceans that we talked about on Monday. So they have a multi-purpose leg, an articulated leg, but not one that can also have a nice respiratory plume on it at the same time. Their gas exchange occurs by spiracles and trachea. They have an expansion, a digestive caecae in their gut, and they have ectodermally derived Malpighian tubules. And this is all about waste reclamation, waste voiding and reclamation of water. And we've talked about Malpighian tubules in a couple of cases before, and this is potentially or likely a convergently evolved trait associated with a move to a terrestrial environment. They're gonochoristic, direct and indirect development, and we'll talk a little bit about some of their developmental pathways today. We're going to talk in general. There's some narratives. We're going to finish with a Guelph narrative about one tropical lineage of insects that lives in Guelph that you might not be aware of, depending on how long you've lived here or whereabouts in town you have lived once you're here. We're going to talk about flight. We're going to talk about development. We're going to talk about sight. Again, we've talked about sight before, but there's some neat things to talk about. So here we go. I think one of the things, so there is my personal bias about this. Like, I am excited by this group and I get to study them. These are all groups photos that I've taken and groups that I work with in and around on campus and in Costa Rica. There's 6 pictures there. 4 of those are from Costa Rica. 2 of them are from the dairy bush. So the ants on the outside, this Lasius with that egg, and this Formica ant that's tending that leaf hopper, those are from the dairy bush. And then we've got some army ants, a tremendously Elton John style flamboyant weevil, a fly whose eyes just baffle me, and we'll talk about some of those eyes and the ommatidia and the different shapes and different wavelengths that they might be receptive to in a second. This fly, for instance, this has a name. These 3 do not. And then this is a colembla, a springtail, and this is an entognath, and so that's one of the hexapods we'll talk about, but we won't have enough time to jump into a lot. But I wanted to share with you these colembulans because sometimes people aren't aware that there's a 0.5 millimeter long little muppet that has purple freckles walking or living underneath their feet. And so there's my bias that I convey to you and try and share with you my excitement about these things. But why else might you want to be excited about hexapods? And it's these calculations that I read in your text. So you might have read this in the chapter that talks about hexapods. I think it's 22 or 24, where it starts out by saying there's something like there's this many, 200,000, I think, insects for every person alive. And that calculation's way too conservative, like way too conservative. Based on the current number of people alive on the planet as of today and the current estimated number of insects, there could be as many as 200 million individual insects for every one of you on the planet. And contrast that with birds. Why would you want to study birds? They eat insects. I mean, that's interesting. But there may be 60 birds on the planet for every person. So inevitably, you're going to have to share your birds with somebody else. And I suppose that's an interesting thing, that if you're seeing a bird, you can think about who else has seen that bird. But chances are, if you are sharing some personal and alone time with an insect, with this ant, for example, or this fly, or this colembula, or this weevil, or this army ant, or this Lasius, the chances are that I was the only human being that saw them. That's kind of cool. There's a lot of these things out there. They're the most diverse things, the most abundant things. We can't help, but we ought not to help, but be excited about these things. So within this gibré and edgecomb phylogeny that we've been kind of marching our way through, dealing with the eucalisterates, the myriapods, the crustaceans, or really, let's call it part 1 of tetraconata, these four cone-bearing eye-like things that make the unified clade of hexapods and crustaceans. We're going to zoom in to what is a good, solid clade of hexapods. So these are the entognaths and the ectognaths. That refers to the gnath, if you remember from your words earlier. It refers to jaws, ento in this case, and ecto, external and internal. So you can think recessed, kind of hidden. That's the colembula, the proturans, and the diplurans. And then we've got all of these insect groups that we will talk about. So my summary of that little plate in your text really has to do with the apomorphies for the hexapods, that we have the division of the trunk into, so we have three tagmata. There's a head, a thorax, and an abdomen that we see. Yes, so the chat is up. Hexapod is a monophyletic group. It is a good group. Good group. They have three pairs of legs, hence the name. They're adults and usually immatures that we call juveniles, mostly with two pairs of wings. The legs and the wings are always on the thorax, so the abdominal segment and the head segment is without legs or wings. One pair of antennae, a tracheal system, and gonoducts and malpighian tubules as excretory organs. So 3 pairs of tagmata. You're thinking, oh my God, I hate this word. This drawing that you've all been able to do since kindergarten or earlier of an ant, when you did this ant and you drew 3 circles and either 3 pairs or 6 individual legs or an antenna, you're drawing an insect. You're drawing a hexapod, and you're showing specialized body segments. You're showing the head, the thorax, and the abdomen. So well done you. Well done you historically back in kindergarten when you began to draw ants and people looked at you and patted you on the head and said, that's a very good drawing of ants. You should go into biology at the University of Guelph. So this is your, from chapter 22, good for me, I did remember it. This is looking at different orders of insects, and we have these kind of, they like the bracket system on the outside to show you these groupings across. We're going to talk about some of these orders in particular. So we've got hexapod there, pancrustacea or the tetraconata. Insects as being a good group within once you exclude the entognaths. And then we've got Polyneoptera. That includes a lot of Plecoptera, orthopterans, groups that use so let's say, so Ephemeroptera and Odonata. So these are kind of paleopterans, they're called, but so Odonata are your libellule. They are dragonflies and damselflies. Ephemeropterans, your mayflies. Plecopterans are stoneflies. In here, you've got some smaller groups that we're not actually going to talk about a lot today, but lots of associated, so sucking things, your hemipterans. And then with the Holometabola, that's a particular lineage where the vast majority of insects lie. And we'll spend some time talking about what makes a holometabolous insect a holometabolous insect. And there's a different, this Sasaki clade phylogeny, I like it because it shows you, instead of kind of wowing you with the words, and like granted there are a lot of words there, but you'll see you're becoming, so there's hectopods, there's ectognaths, there's holometabola. But I think in showing you the colors that are here and the colors and the shapes of the insects, I hope you become somewhat reassured that these are names that you already know. This is not kind of learning together about cnidarians or xenoturbellans for the first time. This is thinking about, okay, Hymenoptera, I know that. That's your bees and your ants and your wasps, and your dipterans, these are your flies, and your coleopterans, these are your beetles. And so these are names in here that you ought to be familiar with already. And so what I'll do is kind of explode this clade out, and we'll talk about some of the defining features for each one of them. So we'll start down here with the entognaths, and this is our homage to the collembola. That's really the group that will spend not a lot of time with the entognaths, but that's the one that I'm gonna ask you to imagine. So the mouth parts, entognath, the jaw parts are sunk in a depression in the head. They have no wings. They are ametabolous, so they don't follow the fact that smaller ones look like the adults. This is ametabolous. So immatures in holometabolous insects, the immatures look very different. Imagine caterpillar and butterfly. So in this case, they're ametabolous. So if you find a tiny little collembola, it might in fact be a different species, but it might in fact just be a young collembola. They lack flight and they lack efficient mechanisms to avoid desiccation. So the ectognath, this was all of the insects writ large in a strict taxonomic sense. So the mouthparts are not recessed in a pocket on the head, and they have compound eyes and ocelli. The neopteran, this is the large group of the vast majority of the diversity of insects that can flex and fold their wings over their abdomens. So it's not dragonflies, not damselflies, not mayflies. Those are earlier groups, earlier lineages, or other earlier branching lineages. And then the Holometabola, these are parts of neopterans. This is where you see beetles and leopard butterflies and moths and flies and bees and ants and wasps. So, and the principal defining character here is that the immatures and the adults are morphologically and ecologically very different. And there's lots of reasons that that's likely been quite a successful, that there we see most of the diversity on the planet residing in this holometabolous lifestyle, and it's because it permits resource partitioning, and it avoids having juveniles compete with adults for the same resources. So there we are again. So there's recessed and ametabolous, so small ones are younger. Then we have the ectognath, so insects writ large, so that includes early branching insect lineages like the dragonflies and the damselflies that can't fold their wings. And then we have the neopterans, which includes all of the kind of younger insect lineages that can fold their wings. And then we have the holometabolous insect lineages, the vast diversity that can hold and fold their wings and have larval stages that don't look like the adults. So, I gave myself this reminder to verify le chat, and I see in the chat right now that there is no question more current than confirming that hexapods are a monophyletic group. Yes, hexapods are a monophyletic group. So we continue with the entognaths. So these are some videos of Collembola, and if you didn't know, if you haven't seen Collembola before, you live with thousands of them. Millions of them. Like if you go out and lie on your lawn and be low and slow, and eventually you'll see little things moving, almost very fast, almost ephemerally and jumping. And they might be globular. This is a particular lineage called a globular, a Smith and Sturrid Collembola. But they have, but they can be longer and darker. If you've seen snow fleas in the wintertime, I don't know what that sound was. If it was me, and if you're not here anymore, tell me. He regains his trail of thought. Snow fleas, this black, like his cross-country skiing or downhill skiing, snowboarding in the last kind of days of the spring, and you see dark little like pepper flakes on the snow. Those are Collembola. Those are protoneuron Collembola. They have the absence of wings, the absence of Malpighian tubules. I understand what it is. They have an amazing adaptation, two of them here that you can see. They have a colophore that allows them to counteract their thin exoskeleton that allows a lot of water to escape. And that is counteracted by the colophore, which allows them, kind of comes out of their belly and allows them to absorb a lot of water very, very actively. So you find them generally in wet spaces. And that kind of popcorn-like motion is accomplished by this organ here and here, which is called the furcula. And the furcula is something that extends up under, I don't think I have a video of this coming, but it extends, this is extended because when I captured and killed this particular one, or when it passively fell into my trap in Costa Rica and it died, it had sprung its furcula. So to be actively, it would be cranked up underneath the body and attached, kind of imagine it by your sternum where it would have a little trigger. And so it would be walking along, and then when it needs to move very quickly, the trigger is with the segments of the body kind of released, boom, and that fires, and fling, you have your spring tail. And there's the colophore there. So lineages include the Collembola, the protoneurons, and the diplurans, and we're not going to have a lot of time to talk about a monocyclic. I'll have to check. I think it is monocyclic. So I've put in an extra. No, I'll check. I don't think I've misspelt that, but the sound in my room, the sound in my head, it's distracting me. So I'll check the spelling for that, Amber. Can it control the direction of the jump when using the furcula, or is it just a quick response? It is, in fact, a very not controlled response. If you Google, there is some increasingly high-resolution, high-speed cameras that have filmed collembolas moving, and it involves a whole bunch of like bouncing into something and then landing and then doing shaking it off and kind of moving around. So there isn't a whole bunch of control that they have on it. It's just away. So quick response, get away. So now, to the ectognaths, defined by these external jaw and mouth parts, and for the hexapods in particular, or for the insects in particular, this is where we're going to talk about a lot of these particularly amazing mouth parts and how they're used in different lineages. So the ancestral one, remember with the mollusk, we talked about the ancestral, what we imagine the ancestral mollusk to be with its radula and kind of rasping across marine environments. So in this case, the ancestral hexapod mouthpart, we imagine it like, imagine a grasshopper. So like cutting and chewing. That's kind of the ancestral use of the body parts, where of the mouth parts, where the mandibulate is hard and heavily sclerotized, so it makes these nice cutting jaws. And that basic ground plan has been modified in a bunch of different lineages, so that we now have carnivores, parasites, parasitoids, modified for liquid diets. So we have the maxilla here up in figure E there, where we have, you can see the mandibles are quite small, and we have nice labellum, which are kind of for this is a biting fly, so it's for sopping up a liquid diet. Then we have labial palps and long maxilla in this Lepidopteran, with a nice kind of coiled up, retracted and protected long fire hose of the maxilla. And then we see here, with another fly lineage, in figure A, the labrum being large and extended, maxilla and mandibles not as very extended and tiny, almost like precision scissors. So even though they can be carnivores, they can be parasites, they can be stylates associated with blood sucking or fluid sucking. That doesn't have to be blood; it could be into plants, of course. Herbivory is the most common lifestyle associated with hexapods. And in fact, hexapods, even though when you picture herbivores, and you're probably thinking browsing ungulates, for example, but the most significant herbivore group in the world are hexapods, are larval holometabolous insects. So in tropical forests where I work in Costa Rica, up to a quarter of the leaves that are above in any stage of the canopy, 10% to a quarter of them, will be consumed by insects. And phytophagous insects, so ones that eat plants, comprise nearly half of all insect species, so 43%. So, and that diversity reflects what has been, through evolutionary time, a coevolutionary arms race with angiosperms. And I'll show you, I believe I've got a phylogeny that I made the cut into today's lecture about hexapods, where we were going to talk about what stimulated what. And so basically the hypothesis that's coming down the pipe in a second is that there is an explosion of angiosperm diversity, and then a subsequent explosion of hexapod or insect diversity following that. Now, before we get to that, I want to talk about flight, because hexapod flight is, again, when you're imagining flight, you're probably thinking birds, because you think about birds. But the first and the most abundant flying animals are insects. And this is kind of an amazing way that they've done it, because it is unlike how bats do it. And it's unlike how birds do it. It's unlike how any of the other flying reptilian or dinosaur-like dinosauroid lineages did it. And they do it differently because the wings, hexapod, insect wings, are not appendages. That's kind of amazing, right? The appendages of this insect, of those beautiful, of that beautiful parasitoid or those beautiful monarch butterflies, the appendages are the legs. What are the wings? They are not appendages. They are outfoldings, like muffin tops, of the body. These are thin folds of the body wall, and that's kind of that's a unique, and it's been it's maybe strikingly inefficient way to fly, but it's been incredibly successful. Now, it was successful from an evolutionary perspective right away because it gave them an advantage over strictly terrestrial invertebrates, that they could disperse, that they could escape from predators, and rapid access to food and other habitats. And then, furthermore, within the neopterans, the lineage that can, like most, so excluding dragonflies, damselflies, those kinds of things where the wings can't be folded. Folding the wings let them have the best of both worlds, that it let them kind of return to leaf litter and in the soil, but then also unfold, boom, inflate their wings, their body wall, and let them kind of fly away. Now, so that's, to me, I remember learning that to me was like the first kind of mind-blowing thing about insect flight. The second way is how they fly their muffin tops, because they do it indirectly. So what we're looking at here, this is a direct flying, that video is a direct flying dragonfly. So there are muscles in that lineage that are pulling directly on the wings. This is not the way that most insects fly, either in terms of abundance or diversity. These are two kind of very old lineages that do it this way. Most insects fly indirectly. And so we have these tergites that are connected. So these are segments of the thorax, so hardened exoskeleton, hardened cuticle, that are connected across by these longitudinal muscles across the body from anterior to posterior, and then the dorsal muscles from top to bottom of the body wall. And what we see in flight, indirect flight, that most insect lineages use, is that the wings, the muffin tops that are supporting them in the air, the muscles aren't connected to the wings. The muscles are connected to the dorsal and ventral parts of the body cavity. So your body cavity, this is, or your external skeleton, think of it like you've got two of these lids of some pasta sauce, and you can pop them like that, right? Okay, so that's the elasticity of having kind of that recurved body wall. And what's happening is that the muscles on the inside are pulling on that. So they're essentially pulling on this clicker toy and flapping the wings indirectly like that. So the upstroke, the depressing of that tergite by contracting the vertical muscles, and then a downstroke, arching the tergite by contracting the longitudinal muscles. So it's almost like this elegant it's contrasting muscles that we talked about a lot with circular and longitudinal muscles when we talked about hydroskeletons. But this is indirect movement caused by contrasting and conflicting muscle systems firing on indirectly on the body, on the thorax. And so this is indirect flight. And there is a little GIF showing you how it's doing it. So you've got these the red muscles in the center. There are the longitudinal muscles that are pulling it, and in contrasting with the dorsal and ventral. And I think that's amazing. So you're flying with your muffin top, and you're doing it by firing those muscles on your body cavity. That's amazing. Well done, insects. Just in case anybody else's mind was blown. All right. I think there's a vestigial question about Entognaths and Collembola. Is that furcula firing just for escape? Generally, but yeah, generally, I would say it is for escape. So then we've got the question, why would winged insects that can't fold their wings not be able to return to the leaf litter? Principally because if you imagine the dragonfly, so for instance, I can show you this here because I can't turn to show you what the dragonfly would look like getting into the leaf litter because I've got nowhere to hide my wings. So it's like hiding a great big long thing instead of folding your wings up nice and tiny and protecting them. So there's the space that it would take, and then also the damage that taking an exvagination of your body wall and trying to push it through rocks and twigs and decaying matter, the damage that it would cause to your body wall. Why did the folding wings allow new habitats? Folding of wings allow new habitats because, and it's simply because they could both disperse between habitats because they can fly. And once they arrive, I'm picturing in my mind like a tiny little leaf litter beetle that might need a particular set of conditions or a particular kind of a plant or a particular mountaintop. It can then move between mountaintops conceivably. It can get into the air currents, move around, fall down again, and then find a new place. And that the other ones couldn't. If they can fold but not fly, they can't move between mountaintops. If they can fly but not fold, then they can't access that habitat terrestrially. I hope that explains some of that. We continue, and we move on or move away from talking about flight, which has blown my mind, and I'll use my vestigial unblown parts of my mind to talk about the eyes. And we've talked about hexapod eyes a bunch in terms of ommatidia and these lateral ocelli, like looking in this particular fly. At these, so the single eyes and then the thousands, potentially thousands of ommatidia. And the wide range of ommatidia we talked about before, meaning associated with kind of a pixelated view of the world. And what we didn't talk about the fact was that most of them, were very sensitive to different wavelengths of light. And so we can see phototaxis within the hexapods that's often cued by wavelengths of radiation that is not within our ridiculously limited vertebrate view of the world. So we think about phototaxis when we've talked about it, so response to light. We talked about it between light and dark, or shadows, into some of the very simple, just eye spot bearing things. And then when we think often, and this is a bias that I think we're all going to be guilty of, when we think about responding to light or phototaxis, we're thinking clearly about the wavelengths that we're seeing and being manipulated or responding to those wavelengths. But when you can see non-visible wavelengths, when you can see into the ultraviolet wavelengths, there's a whole bunch of signals that the world can give you, like this buttercup, as we see it on the left, compared to how the insects see it, with basically a landing strip with concentrated color saying, focus here, this is where the stamen and the pistil are, this is where you get some return and where I need you to go. So this is some view, a new view for us of the intimacy of this association between plants and insects. And they're doing it literally under our feet, and they're doing it kind of in a, if you're fans of kind of Marvel, I'm thinking about the multiverse. This is kind of a multiverse in the natural world, because these are things that are going on around us. These are wavelengths that are here. We're just unable to see it. So this is the insect and plant ultraviolet multiverse. And so these amazing eyes, this is a view of your housefly that's probably sitting, there might be some dead ones sitting on windowsills right beside you. And this is breaking up the eye of that particular housefly and saying there's different ommatidia. So we have the idea, the understanding of a pixelated view of the world. But here, it's a fact that these are complex arrangements of ommatidia that have different spectral associations. So we have wavelengths and different colors or different wavelengths that are received by different parts, different sectors of that ommatidia. And I think that's amazing. That's kind of the theme. Is there a theme for today's lecture? It's hexapods. That's amazing. Now, I can take you, because we can expose some things to ultraviolet light so that we can kind of share how they're seeing it. And these are cabbage white butterflies that you're going to see, if you haven't already seen them, you've seen them later on in the summertime. These are males and females, and they're a nondescript-ish butterfly. They move around. This is how they see the world. But consider when we expose them to ultraviolet radiation and we think, oh, from our bias as a vertebrate, we think there's no sexual dimorphism until we see them under ultraviolet radiation, in which they can see. And we see that, in fact, females reflect far more of that light. And so the males and the females appear dramatically different. And so when we kind of wander around with our great authority and say, well, they're not actually sexually dimorphic, it's like, well, hold the phone, vertebrate. They are. So this is us looking at the world on the left-hand side, males and females, and then under ultraviolet, where the female reflects much more of that ultraviolet radiation and kind of looks how we would imagine it according to our bias. But the male, not reflecting it, becomes a much darker thing. And so seeing the world is kind of a crummy, even for our own species, because of the different challenges that members of our own species have with sight, extending from lack of sight to color blindness, whatever. It's a crummy synonym to understand. To see equivalently equaling to understand is ridiculous, because I hope what this has just convinced you is that there is much more that we don't see than we do see. So if we mean to understand, we should just say to understand. That's my soapbox for you today. And we're going to move on and talk about the Holometabolous, the amazing insects. Now, so we're going to clear up the fact, or reiterate the fact, what I said earlier, that in the ametabolous orders, the young are identical to the adults. So we see that in the Collembola. And then with the there's a division called a hemimetabolous. So juveniles lack wings. And that's like the dragonflies. So dragonfly larvae are aquatic and they lack wings. And they do not resemble the adults. So mayflies and dragonflies. In the porometabolous, like grasshoppers, the juveniles lack wings, but they do resemble the adults. So a small grasshopper might be a grasshopper, but it won't have wings, and that's how you can differentiate it. And then the truly holometabolous, where there are juvenile instars, pupa, and imago. That's the image that you've been seeing looping through here, that you're familiar with, thinking about butterflies and moths that maybe you've raised yourself on your kitchen counter or in your bedroom. So instars being significant changes in the larva that are not associated with terminating as an adult, but basically become bigger. And so we think there's lots of literature about different lineages of butterflies and moths with different caterpillars essentially becoming bigger and bigger and bigger. And those molts are associated with changing in size. We've talked about molting before. We're going to come back to it in a second. We're going to expand on it in a second. And then pupa, that's that last stage, the kind of the cocoon. So it's a non-feeding stage, quiescent. So like and that's where the metamorphosis happens into that final gorgeous. I'll acknowledge that. I don't have much of a love for lepidopterans. I figure there's enough people who have a love for lepidopterans, butterflies and moths, that I don't have to preach to that crowd. But yeah, they're gorgeous. And that video is amazing. You can go and watch it later on there. So that process of metamorphosis is under endocrine control. And we've talked about molting before. And so the particular ecdysiotropin that is going to be secreted by the protocerebrum to the corpio cardica. This directs the corpio allata, which is just nested underneath it. These are in the brain, in the head of the insect, to synthesize juvenile hormones. And the juvenile hormones, that directs down here, the protothoracic gland to release ecdysone. And this is what we've talked about before, this molting hormone. And remember, so that's what initiates a new molt. And so there on the bottom, I've flipped this image, 22.4 part 3 from your text, looking at, for a holometabolous beetle here, moving from an egg to a larva, different instars, you can see, so it doesn't really change form, but it gets bigger and bigger and bigger. Then we have a quiescent pupal stage that finally transforms into the imago, or the adult stage. And we can take a look at that in a different layout here from Rupert, Fox, and Barnes, using a suspiciously similar image. And that stage transition between different instars, pupa to adult, so egg to larva, larval instars, larva to pupa, pupa to imago, that's controlled by the amount of hormone that is released. So if the juvenile hormone is high, the molt will be larva to larva. If it's an intermediate, so it drops down the level a bit, it'll be larva to pupa. And if low, the molt will be from pupa to adult. And if there's none, that's when the adult characteristics can develop. Now, one of the crazy interesting things to me about this larval stage, I see somebody waiting in the room. Hello and welcome, or welcome back, is that as we're moving through these larval stages, and even here in a pupal stage, that as we see we'll see the development of adult characteristics, like so the Malpighian tubules and wings and the exvaginations of the body wall that will be pumped up and filled. So the wings and the legs. And that we have, you can see here, the guts transforming through time. So day 1 is on the top, and day 13 is on the bottom. So time goes top to bottom. So we see the expansion, an increased capacity to breathe, and a contraction in some cases of the gut. Now, put in the chat for a second why you might see the contraction from the caterpillar stage to the adult stage, why the gut might get smaller. Any ideas? I'm seeing some of you, now that I'm in the chat, I'm seeing that some of you couldn't hear. So tell me, I think you can still hear me because there are some answers popping up. So I'll come back to some of those questions, or you can repost them later on. So why might you see a contraction of the gut? One of the hypotheses is lighter for flight. That's a good hypothesis. In this case, it's not actually correct. No need to store energy for metamorphosis. Kind of yes-ish. You're right. They don't. But you might imagine that now, as an adult, they're going to need a lot of energy to make eggs. So they might still need to consume a lot of food. Not all adults do, and you're probably aware of that. But some adults will still need to put away energy to food stores to make eggs. Make room for development. Many adults don't eat, so yes, you clearly are. Some of you are. Different food source as an adult. So that's the key. So in this particular case, with this lineage, this is a herbivore up here at the top. So herbivory, we've seen it associated with large and complicated gut structure. Now, as you become an adult, in this case you do eat, but you're basically eating sugar and liquid reserves of plants, of flowers. And that's easier to digest. It's a higher energetic content, and you don't need the same surface area to accomplish that digestion. And so that's what we're seeing there. Now, associated with metamorphosis, there are big changes in respiration here. Now, those are associated with energy, that flight takes energy, and so the muscles that are being kind of formed here, they're going to need a lot of oxygen in order to do their job. And we've talked about breathing before. We've talked about spiracles. I've made the analogy before to those of you who enjoy thinking about plants that insect spiracles are like leaf spiracles. And in fact, you can see in this caterpillar here, that kind of oval shaped, that's a spiracle. Like that looks like a plant spiracle. This is clearly a convergently evolved thing to kind of close off an air opening that if you're concerned about losing water. And what that GIF is kind of breathing through is letting you see breath proceed into the body of this grasshopper. So the spiracles and connecting, opening to the external environment, and the trachea branching out through the body. This is a diffusion-dependent way of respiring. And that means way back to the start of the year when we talked about surface area to volume ratio, remember we talked about the fact that you never have to be worried about giant ants or giant grasshoppers or giant insects coming out of B-movies or nightmares to harm you. And it's largely because of this system of respiration that they couldn't possibly respire efficiently enough. They're a great, respiration at this scale is great for them at this physical size, but that's it. So this is essentially the syringe model where so take a look at this kind of engineering drawing at the bottom, that when we see the muscular pump that you've seen probably insects respire, where they use their body, their segments, to pump in and out like a bellows or an accordion. And that muscular pump is moving and pushing air and hemolymph, which is the blood, together and making or facilitating that kind of passage of air into the body. So it's drawing, that pumping action is drawing the air in, and it's also then kind of undulating the blood and mixing up the blood and the air to facilitate that transfer of oxygen. Now, the tracheoles these long filamentous, the lungs, another exvagination of the body wall, they're lost during molt. Now, I want you to think about that for a second, and I want you to kind of think about it. I will rarely encourage you to anthropomorphize the wonderful things that we're living with here, but in this case, I do want you to think about it. Molting is like ripping your lungs out. So if all of your tracheoles are lost during a molt, that's very stressful. And that's likely going to be compounded as ambient temperatures rise, because that transfer of oxygen is dependent on, can be affected by the temperature in which we're thinking about. So this picture that I took of Jack, our son, and this molting cicada, where you can see this, the tracheoles, the old tracheoles extending out of the exoskeleton. There's a period of time of about, it can be upwards of an hour, where essentially this molting insect has to hold its breath. This one has already passed through it, but there's where you're ripping your lungs out to transform. And so what I'd like to do in the last few minutes here, I'm hopefully going to get to my termite story, but what I want you to do is kind of use your stilo for a second, take out your annotations tool and plot the difference. So you've got two larvae, maybe use a red for a molting larva, one that's going through this rip your lungs out, and a non-molting larva through time. So they're both, so the level of ecdysone is different in the two of them. One of them is molting and one of them is not through time. And we're going to see what changes happen in oxygen consumption. And the difference between the red and blue plot is really what's happening when you've ripped your lungs out. So interesting. I'm liking this. I'm loving this. This is awesome. Okay, so we've got some, let me, just so that I don't forget, I'm going to grab a bunch of those. Okay, stop drawing for a second. Please, thank you. Put your hand up in the air, pat yourself on the head. So we've got molting larvae, for instance. So the idea here is that the consumption rate through time, it should still go up. So you're indicating, I think, the success in what you're indicating is that it's not being interrupted. But remember, if the oxygen consumption rate, it will change through time. It'll tend to go up. But you're not seeing a break in it. So that is correct. Now, what you're seeing or what I'm seeing you say in red is you would expect to see, some of you thought catastrophically down, some of you thought up and then down, and some of you thought down and then up. And that's super interesting. I'm going to take away your drawings for a second here. Thank you for them. They're gorgeous. And this is what they actually, I have to get rid of mine, saw. So molting in this case is red. And so the natural non-molting is just kind of a linear increase. And what we see, though, interestingly, and this is new information, this study's only about like our understanding of how molting affects the trachea or the tracheoles and their capacity to respire, how stressful it is a relatively new bit of information in the 21st century, is that their rate for molting larvae, they know it's coming. This is another consequence of that pumping up or change in the molting hormone, is that they essentially, just like a deep sea diver before a free diver, they out-respire a non-molting larva. They're clearly trying to charge up their body. And so it could be up to, in the paper talks about a 45% increase in oxygen consumption. And then catastrophic stop. But then as soon as the tracheoles are engaged and hardened in the new body, boom, it's way up at the top. And so so you're right if you talked about a ramping up, a catastrophic stop, and then compensation. And so there's kind of two periods of compensation, one before molting, interruption, and then compensation afterwards. I think that's amazing. I'm going to remind myself to look at the chat here in the last 3 minutes. Okay, we're going to talk about sociality in the last 3 minutes because it's a thing that I want you to think about for over the break. And think about it if you're staying around town and socially distancing yourself and thinking about tropical social insects. So social insects, ants, bees, This is when there's a reproductive division of labor. So we see males doing one thing, queens doing another, and then we have sterile castes, so overlap of generations, and so workers and wingless queens that do different things than do the queens. And so if you imagine a bee colony as you looked at earlier for the finding, or the ant colony that you're looking at here in the dairy bush, these are truly eusocial insects. Now we have bees and ants. We have also a non-hymenopteran lineage of social insects in Guelph. We have termites, and I have 2 minutes to tell you the termite story of Guelph. This perhaps is news to you that we have termites in Guelph. They look like this. We talk about them in the news and in real estate in this city quite a bit because there is a war on termites. This is a screen grab from the Mercury or Guelph Today, what it became a few years ago, celebrating the fact that there are fewer and fewer of them around on city plots. We have them in Guelph because of this particular mill, Goldie Mill, which is on the Speed River just north of the downtown. And once it stopped milling itself, it was bought out by a conglomerate that used it basically as a place to store wood from all over North America. And so probably what happened is termites arrived in one of those shipments from the south, and then they stayed. And we haven't had cold enough winters to destroy those populations since then. So we now have southern populations, a disjunct southern population of termite that lives in Guelph. And we're not alone in this. So all these dots across southern Ontario, so in Toronto, York, Scarborough, Elora, Fergus, Dundas, Windsor, down into Peely, we see these termites and we see the fact that they can survive. People have done critical temperature biology upon them to figure out whether or not a nice cold winter will freeze them out. And as long as they can bury themselves, the evidence is clearly no. They've been here for decades and decades and decades now, and the populations, we can fight them back, but we have not eradicated them. And so we see here different very high temperatures. So if we get hotter, they're likely going to be fine with it. And the minimum temperatures, you might think, well, that's not a very low minimum temperature, but it's fine as long as you're underneath the frost layer. So we have a professionally a PhD holding entomologist as part of the City of Guelph employees. We have a termite control officer, and then we have these regions across the city, red and blue, that have seen, this is 6 years ago, back in 2015. Red is where we have found active termite colonies. Blue is where there are adjacent to that. The yellow is my selfishness going, that's where I live right now. We can see 2018, some of the areas get eradicated, some appear new, and then this is last year where someone has clearly moved termites around in the city. And so it's 20 past. That's the end of this week. Please take care of each other. Have a nice, responsible, help us avoid a 3rd and 4th wave of COVID Easter break if that's your druthers. But get outside and do good things for your brain and your body. I'm going to hang around for a little bit on the chat. If you've got some questions that I haven't answered yet, I'll try and do that there. And I will otherwise see you on Monday. Take care of yourselves.